2021. It's the last day of March. It's March 31st, which means April starts tomorrow. And the president has given us all 30 days to get our act together and destroy this virus. So here's how we're going to do it for anybody who wants to try to do it this way, which is actually stopping the actual virus. So I'm in Foldit right now. Go to foldit.org. Just go to Foldit, download their software, learn how to fold proteins, go through their tutorials, and become the savior of the world. Become the next Madame Curie of 2020, and figure out how to fold proteins. Now, what you're looking at here is a way of looking at what's called the ORF6 prediction protein. We don't even know what it looks like. It's called ORF6, and it's part of the coronavirus. It's part of that spiky ball sphere thing that you all hear about. Well, this is a way to look at it with a bunch of hydrogen bonds. Well, here's where I can help out. So this is the COVID genome sprap thing that I'm building in Microsoft Excel. It's a way to look at the virus in a mathematical table type of way. So what am I talking about? You don't have to follow along, but it's fun if you'd like to. Okay, these are all the genes. This is the order of the genes. This is how many nucleotides or base pairs are in these genes. This is the Italy version, a strain that was taken in Italy in, I believe, December. ORF6 is what we were doing over here in Foldit. This is a puzzle to predict what the ORF6 protein is going to look like. Well, if you look at the order of all the genes, and there's like some bumper DNA that's in between the genes that may not code for stuff that's small amount of base pairs and stuff like that. Well, there is an ORF6 in there. Where is it? It's right here. It's in the 7 spot when it comes to the order of categorized genes and things. The way we can look at it is by clicking on ORF6. Actually, MORF6 is the beginning of ORF6 and M, and then the actual ORF6 is right there. So what this shows you now is that pretty far down in the genome of this virus, there's this beginning part, which they say doesn't do anything, even though this part actually starts with an initiator codon. And then it goes down to here, which is what they call ORF6. So ORF6 is all this stuff. And what the colors and everything, what you're looking at, and why I think this is going to be fun, is this thing right here, which we're supposed to fold around and model and see how this thing's going to fold up and bond. And this is like a sample way they think it's going to look already. You can look at this thing without showing hydrogens. You can do it, and instead of sphere, you can do it in a different type of way of looking at it. Here's like a nice asparagus way of looking at it. And these are like hydro bonds or something. I don't know what everything means yet, and I don't have the genome here in order, but I kind of do, and that's kind of what I want to talk about. So what is this asparagus helix? It's like a fusilli or a rigatoni and a stick. Stick you pick off the ground. What, with all the stuff sticking up, what is it going to fold into and look like? Well, let's get rid of the MORF6 part right now and assume that this doesn't matter because this is some type of initiator that doesn't code. Probably because this is like you see a bunch of things that are polar or hydrophobic. This is where we're going to learn together, everybody. Okay. So initiator, this means something starts coding the genome characters. This is the amino acid. We can start on codon 1, which makes this look a little easier because this is just showing the 3 triplet pairs of DNA and what their amino acid is, what the protein symbol is for it. Mass of just one of the base pairs, so don't pay attention to mass right now. Don't look at that. The order of the genome, where this is located. So this is on the end of the genome, which is kind of towards the end. It's about a 30,000 pair genome on the end. This is not too many base pairs. It's only like 62 times 3. It's like 180 or 190 base pairs. It's not too much, but what is it doing and what is its shape? And why do we care? Because it might be on the edge as like a spike or something on the edge of the protein. So we kind of want to know what shape it is. And I'm going to talk about spikes in a different puzzle in a second. But when we just start scrolling down, because this is all in order, I colored these things to mean something. So There's acid, acidic, or basic, like the pH of the amino acid can matter. This is a CAT, that's cytosine, adenine, thymine. Although it was originally uracil in RNA, but it's thymine, I guess, in DNA in your cell. It's basic. It's going to want to get a hydrogen atom or want a bond, and if it's acidic, it wants to shed an electron or shed a bond or something. So we have different colors for acid and basic. This can kind of be formulated as almost like right or left, because if you have something that's acidic, it's going to be pushing, say this is acidic and this is basic. This thing is going to want to push this way. It's going to want to go that way, because this thing's going to want to shed an electron and shoot off like a motor, and this thing's going to want to go and attack something and meet somebody. This one who doesn't like being quarantined. Basic likes going out and meeting chicks, right? That's what I believe the difference between acidic and basic. It's almost like right and left. People who know more information about this, please correct me if I'm wrong, but if that's somewhat how it works, that's why I'm colorizing things this way. Hydrophobic, I made it blue. So yellow means something just started. Blue means that if you have a lot of blues together, you're probably going to want to form a circle or a spiral sort of, because here's why. Assuming this means hydrophobic, which I don't know if it does or not. It might not. I should know the answer to this, messing around with Foldit for weeks already, but I don't know. Things that on the end of them are hydrophobic are all blue, as we saw there. Okay, a lot of blues are going to want to not take in water. And that means that if a bunch of them are in a row, they're going to form a circle, because they're going to be constantly running away from the water. Imagine you're trying to like blast off here, but you hate water. Like imagine you blast it off and you hate air, right? On the planet. As soon as you blast off from your rocket, you're like, oh my God, I hate air, I hate air, I hate air, I want to go away. I want to go away, which means you're going to like shoot off to the left or shoot off to the right. But either way, you're going to shoot off and you're going to end up curling in on yourself, because you're like, oh my God, I hate all this water, I hate all this water, I hate all this water. And then you're going to loop back, right? So when you see a lot of hydrophobics, I believe that kind of means a circle or a sphere, helix type thing, I think. I don't know. Like, look at that. Boom, there's 5 in a row right there. There's 7 out of 8 of these amino acids. That's 21 out of 24 nucleotides are all hydrophobic in their amino acid pair, creating probably a circle. Boom, and then something else initiates. Shocker. How about that? So this thing here is like a big bubble that's almost completely hydrophobic with a few acids. As a matter of fact, when you look at this almost looks symmetrical with the exception of the basic. Like if this basic was a hydrophobic, I would say this is like a perfect palindrome, rigid sphere of hydrophobia, because you've got like a couple acids powering it in one direction and almost like a bridge, like holding it up. I know I sound like a crazy person, but this is what I see in these colors. I see a huge buffer and a motor right here. And then this is like a stability on the end that doesn't want to fight with it being waterproof on the end. It's like a very rigid thing. This string probably means something. The basic looks off there. The basic is what's weird in that to me. So then you move down and you see another initiator here. And then what happens? See how this doesn't look as hydrophobic? It's more mixed with hydrophobic and polar. And basic and acid, it's all over the place. And polar, like random spotty stuff that doesn't look to form a more unpredictable shape as you go down here. Then you got some super powerful acid on the end of it, and then this thing stops, right? This is probably junk DNA down here also, because there's a quick initiator and then nothing, essentially. So it's probably really stopping around here if it does anything. But it ends with a lot of polar things. I don't know the effect that something polar has in this whatever, okay? So this is why colorizing this, putting this order, and then attaching some mathematics over to the right or some formulas that are doing things based on the order as we go down. Like I'll give hydrophobic a value here, and I'll give basic a value or a direction, and then scatter plot this thing to see what it's supposed to do. So that's super interesting what's going on with the file. Here's something else. Let's go look at another puzzle. Instead of looking at predicting just the ORF6 protein, let's look at the big gun, the coronavirus binder designed round 5. where they have a string. I didn't show you where the string was, but they have a string of proteins for this binder. Oh God, things have changed. What's going on now? Oh no, I did that yesterday. I'm just looking at a different view. Okay, that's this thing right here. This gray thing right here is a spike that they've already found. And let's view it in sphere, I guess. All kinds of different things you can do here. So that doesn't really help. Actually, it kind of does. Take it back. Because look at this. You got this fertile ground for bonding going on here. There's a lot of blue and orange and red. It's a hot spot. It's like jumping on a pancake. No. It's like jumping on a trampoline, sort of, right there. Essentially, a tramp cake. It's jumping on both of them. There's all this activity going on here, and you can see all this gray junk over here. They're not talking about bonds on the side of this. So it's kind of like that's a rigid structure. And I've always said this thing poking out here is weird. This is a weird shape. It's not a perfect, like this thing pokes out a little bit. I bet you that's what's causing the problem. Anyway, so that's a cool way to look at that. Well, what else can you do with this? And why am I bringing it up? So this is, you're trying to bond to this thing. Well, there's a genome of that already mapped. It's called the blueprint. The blueprint happens to be the order of the amino acids. And ADSV, ADSF. So I don't know which gene this spike is on, but I wonder if we could figure it out. How would we do that? I've been thinking about this, but I haven't come up with a solution yet. I might just turn off the video, but let's try for, we're 12 minutes in, Jesus. All right, let's see if I can figure this out in under a minute. If we get to 13 and I've made no progress, we'll stop. ADSVF. So what I'm talking about is this AA1 state. There's going to be an order where it's AD what? ADSFV. ADFSV, but I don't have all those in order right now. But what I want to do is I want to actually put those in order. So I'm going to move them. I'm going past 13 minutes. We're having fun. I'm going to show you how, like, I'm diagnosing and just like all the things you can do with this to try to do some quick analysis on this. So we want the AA1. This is the code for what amino acid it is. Okay. So I just collapsed that. And the order that it was out in the blueprint here is ADSFV. So ADS, you can actually just grab this. SFV and just move them up. We want a situation where all these are in order, and can we find that in the genome? So I have a full order of the genome right here, and we would have to say, here's how you do this. Say if this equals A, that's going to take too long, but I was going to string it down so that it finds where this exists, and then it's going to be able to tell us where in the order we are. Let me freaking do it. I know this is boring, but we're just going to code the string out. We're going to say, well, take this and just use the ampersand. You don't have to use concatenate. This and this and this and this. So now you're at 5. Okay. So you've got the 5 protein string on that codon. So now I just pasted this entirely down. And now I'm freezing it because I don't want any formulas in here. And I'm just going to look real quick and say, well, what are we looking for, right? ADSFV. All right. Well, ADSFV. And the answer is let's see how many there are. There are two options. They are, they're a few things apart. So there's one, there's two of them. And the way you can quickly tell what that is just by clicking on the thing here. It's on gene S. In codon start 1. So this is totally gene S. Easy. Easy. Which makes a lot of sense because it's called spike. So I'm able to figure out exactly where things are. So let's just whoop out gene S real quick. Show you what gene S looks like because I haven't actually looked at this yet. So I'm going to bring everything back. I'm going to do a bunch of undos if it'll let me. No, it won't. Let's get rid of that. Let's unfreeze this. Go all the way back here. Start back at the C order. Amino acids, bring it down. Bring up gene name up here. Bring order all the way down here. So there's a lot of moving of the pivot table around just to get where I want it to be and get the colors right back to where they were. And see if we're getting there. I think I need to move state down one. And let's see if that works. Colors, please be there. Yes, okay. So we are on gene S. Boom. Alrighty. And what's happening is this is going straight down here on this gene S, which happens to be 1,274 base pairs long, much longer. Is supposed to be showing this. Part of it is supposed to be showing this right here. And I don't know where it started, but like when you're looking at it, and we do it in cartoon thin or something, that's what it really looks like there. Or maybe, who knows what it looks like. It's smaller than light can see. We're just modeling it, but all those bonds and stuff, and wherever this thing comes from, I would assume that like hydrophobic chains would be like this kind of stuff, and I don't know what creates these sheets. I don't know how sheets are made because that's what they're called, is sheets. There's sheets and then there's helixes and then there's just random lines and junk. So like here you can see we've got a lot of hydrophobics at this initiating sequence here. Then a lot of polar mixed with a little hydrophobic, then a ton of polar. Polar, this alternating stuff, I wonder if this is lines or do a lot of polar things make sheets because they line up polarly, like they don't have a charge and therefore a lot of them maybe create those sheets. I don't know. More polar. Let's look at what this looks like color-wise down there. More initiating stuff going on here around the middle of this. So what's up with that? Because, okay, and I also need to move something else I just realized. Move AA1 up because the stop, right? Oh no, actually no, I'm wrong. Leave that. All right, I'm starting to run super long, I know, but how does this shape all these things? More initiator on 1,000. Here's some more hydrophobic things and a lot of polar in this. Basic, acid. You know, what happens when you stick a bunch of basics around a hydrophobic and an acid? What does this look like? Things that look like palindromes, in my opinion. You know, they look like a copy of themselves. Like you've got basic hydrophobic, acid, hydrophobic, basic, right? This looks like the same backwards and forwards. What does that look like as a shape in here? I'm going to try to answer that by the time I do the next video because this one was super long. But if you want to get a copy of this file, I'm going to start putting this out on the Discord or on the Foldit chat on Reddit or something. People can get a hold of this because we need to be solving this problem right now. we need to figure out how to bond to this and get it out of our system and not have to fight it with ventilators and with with our treatments that are meant for treating severe lung disorder, where we should be attacking a virus because the virus spreads so easily, so transmissible. And this shape, I believe it's this guy right up here, this craziness right up here. This is probably what's causing the bond, the secure bond into our ACE2 receptors for some reason. This looks like a hook, like Captain Hook will walk around with this and cut off trees with it. I mean, look at how sharp this is. There's actually, I want to find out what part of the genome that is, right? This right here, sorry, this right here looks rough to deal with. This is like a huge hook, right? And then there's like a shield in front of the hook, right? Yeah, there's a hydrophobic shield, right there. That's what that looks like. Or at least a flattened shield to this hook, which is right here. Because if you think about how much that sticks out on the edge of the virus, because the virus is supposed to be attached right here. Like this is where the bubble is with the whole virus thing. This is just a spike poking out of it. And this is its worst part of the spike that I think is doing the most damage. And I'm not a doctor. I don't know what the hell I'm talking about. But that's the way it looks to me, doesn't it? That looks like, look at that. From that view, doesn't that look like that is just out trying to dunk on you? So we got to do something about this. We got to bond to this thing. I imagine this is the troublemaker. So anyway, we're out for now. Let us all solve this problem and be done with this by April. I don't think anyone can staying in their house for another month. I mean, it was already been a couple weeks. Let's do something about that, all right? Let's go.